Welcome everyone as you start to settle in. I just want to take a few moments before we get started to introduce Bill Schutt. He is our author that will be giving our talk today about his book, Pump, a natural history of the heart. A few things before I say a little bit more about Bill. If you guys have any questions, please put them in the Q&A box at the end and we will get to them after Bill is done talking. And I think that really covers it about our questions. So in the Q&A box, and we'll get to those at the end. But back to Bill. Bill Schutt is a vertebrate zoologist and author of 5 nonfiction and fiction books, including the New York Times Editor's Choice, Cannibalism, A Perfectly Natural History. Recently retired from his post as professor of biology at LIU Post. He is a research associate at the American Museum of Natural History, where he has studied bats all over the world. His research has been featured in Natural History magazine, as well as the New York Times, Newsday, The Economist, and Discover. Thank you, Bill, so much for being here.
It's really a pleasure. Nice to do something local.
We appreciate your time.
All right, well, I guess I'll get started. I'm a local. I grew up on Long Island, and I was actually a student at Southampton College for 1.5 years before I moved on to CW Post back in the Cretaceous period. I got my bachelor's degree there, then went on to get a master's degree at SUNY Geneseo, and years later a PhD at Cornell University. And this is my latest book. It just came out last Tuesday. And I hope you find this talk for the next 30-35 minutes or so interesting. So as was just mentioned, my first 2 books that were nonfictions were sort of very different than The Heart. And the first was Dark Banquet, which looked at vampirism and blood. And it was really not a stretch for me because I had done a lot of research on vampire bats. And so when I was lucky enough to get an agent in New York City, and my first book turned out to be about blood-feeding creatures. And I was lucky at the time because ticks and well, unlucky for a lot of people, but lucky for me, there were things like Lyme disease out there, and bed bugs were going crazy in large cities, and of course I wrote a lot about vampire bats. And the follow-up to that was cannibalism, and so I got to explore this phenomenon in a way that I've sort of found this neat niche. On one side, you have all this kind of sensationalized stuff, and on the other side, you have this academic material that only people who are really interested in the science, the scientists who are doing the work, would really care to read. And I've always, as a teacher, been one to take complex topics and demystify them and try to put them into language that's not full of jargon. And so when I started writing books, that was really what I wanted to do. So when Pump came along, I was looking for something for my third book, and my editors and my agent suggested that I look for something a bit more mainstream. And I really thought that The Heart was something that had been so well done that I wouldn't be able to go in there and figure out how to do this, how to write a Bill Schutt book about this. And I did some research, and I was wrong, because there was a lot of neat information out there. And I was interested in looking at the heart, how it evolved, looking at it through different animals, and then moving into myths and mythology, the history of medicine with regard to the heart, and how the heart became this cardiocentric structure where everything is based on the heart, your soul, your intellect, your memory, how that came about, and then look at modern and even future therapies related to the structure and to the circulatory system as well. So I was really pleased, and I had a steep learning curve, and there was a lot of weird stuff in there. And I've always been one to try to inject humor and to make and when I taught, it was my presentations when I'm writing, make my books entertaining. And this is certainly not a textbook. And that's one of my favorite reviews. This book is not a textbook, and it's really not. So this is for people who are interested in science, but not necessarily scientists. So a couple of questions. We'll start out with the basics here. What are hearts and circulatory systems? Well, to to give you the short answer, hearts are, first of all, they're extremely variable, depends on what animal you're looking at here. But for the most part, they're muscular pumps. So there's muscle tissue, muscle cells there, and they're moving a fluid around through the body. And that fluid, when you're talking about animals like vertebrates, is blood. And if it's insects, for example, that would be something called hemolymph. And that blood or hemolymph is moving around through a series of vessels throughout the body. Now, why have a heart? Why have a circulatory system? And what they do, and this is pretty well common knowledge, is that this pump is able to move around and transport throughout the body blood which contains oxygen and nutrients and delivers them to the body, to the tissues of the body. And meanwhile, at the same time, it carries away carbon dioxide and waste products and gets rid of those. So the question that I looked at early on in my research is, do all organisms need a heart and a circulatory system? And really the short answer is no, they don't. Okay, so who doesn't need a circulatory system? Well, first of all, if you're small and flat, you don't need a circulatory system. You don't need a heart. And of course, there are some other animals, and we could spend all night talking about this aspect of it, but there are groups of animals like sponges, anemones, corals, and starfish. They don't have hearts. They've got a different type of circulatory system, which we're not going to go into tonight. But let's look at single-celled protists and tapeworms, long and flat. This guy's about 20 feet long, that tapeworm. Why is it that they don't need circulatory systems? Okay, so basically what happens is, if you're a small little guy and you're a single-celled organism, you generally live in an environment, mostly water, where there is a high concentration of the oxygen that you need in the surroundings, in your surroundings. And you have a low concentration of oxygen inside your cell. By the same token, carbon dioxide, which is produced as the cells are making energy or converting energy into a form that they can use, carbon dioxide builds up during that process of cellular respiration and needs to be gotten rid of because it's toxic. And there will be a low concentration of carbon dioxide in the environment and a high concentration in the cell. So if you're a single-celled organism, then basically what happens is that you don't need a circulatory system because the oxygen just diffuses through your cell membrane into your cell. It goes from a high concentration outside to a low concentration inside. So it just sort of, as they say, follows its concentration gradient. And by the same token, if you've got a high buildup of carbon dioxide inside the cell and not much outside the cell, then the carbon dioxide just diffuses out of the cell into the environment. So there's really no need for a circulatory system. So that type of diffusion works well and good if you're really flat or if you're really small, but it doesn't work if you've got any type of size to you. So this is a view where you're seeing skin here and the underlying fat and muscle and bone. And there are thousands and thousands of layers here that we're talking about and millions of cells. And diffusion does not work efficiently if you're big, if you're thick. And so in order for that to take place, you had to get the evolution of a different method to transport these materials around throughout the body. And that became the evolution of the circulatory system, which you see here. Now, you can sort of break this down, the types of circulatory systems and hearts that are in existence in the animal kingdom. First of all, there's what we have, and that's a closed circulatory system. So here's that muscular pump, and here are the vessels, and they're going to carry this blood to the organs, and the vessels get smaller and smaller, and then the material just moves out of the blood into the tissue, or out of the tissue into the blood, and then back to the heart again. That's a closed circulatory system, which means that the blood that's inside of these vessels never leaves the vessels or the heart. Now, and that would be things like, the vertebrates. So we're talking, and there are some invertebrates too, which we'll see in a sec. But this happens to be a fish heart that you see down here in the lower left. All right, and you can see here that the blood never leaves this circuit. Most animals on this planet, and insects being a prime example, have an open circulatory system. So they have a structure, and I'm calling it a heart with quotation marks around it, because there are people who would not consider this to be a heart because it doesn't have the, it's not a card carrying heart because it doesn't have the right layer of cells lining it. In any event, we're going to call it a heart, and say that what happens here is that as this liquid, which is hemolymph or blood, is pumped out, it doesn't move continuously in a loop. It is dumped out into a large chamber called the hemocoel, and it supplies the tissues that surround that chamber with oxygen, with carbon, and takes away carbon dioxide, supplies nutrients, takes away waste products. So these are two very, very different types of circulatory systems that exist in nature today. Now, let's talk a bit about our heart. How does it pump blood? Well, we all know that the heart is made up of muscle, and here's the way that the muscle is arranged. I picked a four-chambered mammalian heart, this human heart, and there are two chambers up on the top. These are the receiving chambers. They receive blood back either from the body on the right side or from the lungs on the left side. And then you have these two pumping chambers, the ventricles down here. But the walls are mostly muscle. And what happens in a mammalian heart is that the stimulus to make this heart contract comes from within the heart itself. This is the so-called pacemaker. So an electrical current or charge is generated here and moves through the heart. And as it does, these chambers, the atria, and then the ventricles contract. And you can see when they do that because the chambers are made up of muscle tissue. So where does this blood go? This is a figure that you've probably all seen before. Here on the right side, here comes the blood returning from the body. It doesn't have a lot of nutrients. It's given those up. It's picked up carbon dioxide here, and then from the right side, this deoxygenated blood is pumped out to the lungs, where it picks up oxygen, then moves back to the heart, to the left side. The left side of the heart contracts and sends blood out to the body. And also down here, you can see here are these vessels that are carrying the blood to the digestive tract, where it picks up nutrients. Okay, so that's pretty much it. I mean, and that's really in a simplified form here, but that's in a sense where the blood goes. All right, so besides the heart, what is it that helps blood circulate? And if you look at a vessel here, a blood vessel, notice that it's got a wrapping of a muscle tissue. And you can think of it as this. If you had a water balloon full of water and your fingers wrapped around it are these muscle fibers, if you squeeze them as these fibers contract, that water in the balloon is going to move. And that's really what's taking place here. So there are large vessels in our body that assist the heart pumping blood around the body by contracting and helping that blood on its way. This is why, and this is really a short version of this, when you hear about hardening of the arteries, why is that bad? Because this is not, these muscles are not working very well anymore. So the heart has to work that much more, that has to work that much harder in order to help pump blood around the body, and that presents problems. Okay, so let's talk about invertebrate hearts, animals without a backbone. And this is someplace where, if you look at it, there's a tremendous amount of variation here, tremendous. And the reason that we believe that there's so many different types of hearts, or lack of hearts, or muscular pumps, or large blood vessels that help to pump the blood, is because these structures evolved many times in the invertebrates. When you get to the vertebrates in a sec, the backboned animals, we think that hearts probably evolved only one time. So there are similarities that we're going to see, whether you're looking at a fish heart or a reptile heart or mammal heart. But in the vertebrates, and I talk about these much more in depth, there's not a tremendous amount of time to talk about it. The reason I showed this here is I just want to show there's this great amount of variation that you see in the animal kingdom. And a take-home message should be that we have this tendency to think that humans, everything that we have is the sort of end-all pinnacle of structure and behavior, and it's just not the case. When you look at something like a squid, for example, which has 3 hearts, 2 of these hearts pumping blood to the gills, and another systemic heart that pumps blood to the body. You might look at that, or you might look at these aortic arches that sort of act like a heart and an earthworm and think, well, ours works a lot better. You shouldn't really think like that, because these things work perfectly fine for the function that they evolved to do. One little bit before we move on here is that if you're looking at insects, they have several structures which could be considered hearts that help pump hemolymph to places like the antennae or to the wings or to the legs. But the other thing that you should note here is that unlike many hearts and circulatory systems, the insect circulatory systems do not, they don't carry oxygen and carbon dioxide. So they are solely for moving nutrients and waste products around the body. All right, so when you look at vertebrate hearts, this is certainly a whole lot more familiar to people. And here are these pumping structures, the ventricles and the atria are receiving chambers here. And here in a fish, you see this is sort of a simple circuit. There isn't this sort of double pump that you're going to see here when you deal with mammals, and this works perfectly fine. Here comes the blood back from the body. It enters into the atrium. It gets pumped out by the ventricle, gets sent to the gills, picks up oxygen, gets rid of carbon dioxide, and then back to the body again. When you look at reptiles and amphibians, they have what is known as a three-chambered heart. They've got a pair of atria, and they have a single ventricle. And people often point to this and they go, well, you know, there's mixing of deoxygenated and oxygenated blood here. That's sort of defective, isn't it, or primitive? No, it works perfectly fine, believe me. This is what we're used to seeing here. This is a four-chambered heart that would be found in crocodiles and alligators, which is what you see here. This would be a mammal or a bird heart. And there's complete separation of these two different circuits. One is solely dealing with deoxygenated blood on the right side, and the other, and blood returning from the heart and sending that deoxygenated blood to the lungs. And then that oxygenated blood comes back to the left side and gets pumped out to the body. So that is what you see in crocodiles and alligators and their pals, and birds and mammals. All right, so when I did this, when I wrote this book, I found all of these incredible stories about animals that I wanted to tell. And what I told in the prologue in the first chapter is the story of what happened in 2014 when 9 blue whales ran aground and they died up in Newfoundland. And this was tragic because that was quite a large proportion of the blue whales that lived in that area. But the key here is that in most instances, blue whales, which are the largest creatures we think that have ever lived on this planet, when they die, they sink. So they were never really popular with whalers because not only were they fast, but if you harpooned them and killed them, they sank. So my friends at the Royal Ontario Museum in Toronto had been working on mammals and especially whales for a long time. And they kept getting these questions like, what's the largest heart in the world? Blue whale heart. How big is it? It's probably the size of an SUV, but they never really knew. And so in 2014, when these 9 blue whales died, 3 of them didn't sink, and they wound up washing ashore up in Newfoundland and in these remote little villages or towns. So these guys decided that they were going to go and see if they could recover a blue whale heart. And that's what they did. So they went up there, they had heavy equipment. You know, we're talking about if you cut into this thing, there was 4 guys that climbed inside of it to push the heart out between the ribs because they wanted to answer this question, how big is this heart and can we preserve it? So this presented a boatload of problems, as you can imagine. And there's the ROM staff. There's my friends, Mark Engstrom, who was the curator in charge. This is my bat biologist friend, Burton Lim. And when they removed the heart, they realized that first of all, it didn't look like a normal mammalian heart. And I described it as, when I saw it, I thought it sort of reminded me of a 400-pound soup dumpling. If you were to go to a butcher shop and get a heart and place it on the counter, it would have this form. It would sort of look like a human heart. But this heart was very different. It literally collapsed. And so the questions are, why? Why is the heart built differently? It has 4 chambers. It's certainly a mammalian heart, but they think that they're hypothesizing now that instead of having this sort of maintaining this heart, this normal mammalian heart shape, that this thing collapses because when these animals dive to great depths, their bodies are under tremendous amount of pressure. And so in order to cope with that, rather than having pressure on the heart from the outside, these great pressures, the thing sort of collapses. And when your heart is beating 10, 15 times a minute, and when you're diving maybe 2 or 3 times a minute, then this is the way that they are adapted to being able to dive deeply. Now, so they removed the heart, they froze it, and then they had to, of course, they had to clean the thing out, and they brought it back to the ROM. Here it is being transported in a large truck. But when we went into when we took science classes, we were always worried about spilling a little formaldehyde on ourselves. Here they were using 1,000 gallons of formaldehyde to preserve these hearts, and so you had to keep the preservative inside the heart. So they had plugged up all of these large vessels with every type of bottle that they could find. The thing that was most peculiar, once they sent this thing off, and it took 5 years to preserve, they sent it out to this plastination facility in Germany. If any of you guys have seen the bodies exhibit, where you've got this guy who's dribbling a basketball and he doesn't have any skin, which I don't really recommend. But this is the largest. This heart had literally been transformed into latex and color coded so that you could see the veins and the arteries, and it was just absolutely stunning. But one of the things that jumped out at them when they looked at it and they finally saw it was the fact that it was a whole lot smaller than they thought it would be. And so the question is why? And the reason being is that if you were to look at something like a hummingbird, and a hummingbird is tiny, of course, but a hummingbird's wings can beat up to 80 times per second. And in order to do that, they're using their muscles. And that requires that these muscles get a tremendous amount of energy and oxygen in order to beat that quickly. So what happens when you're looking at a hummingbird is that the heart rate can be 1,260 beats per minute. That's insane. And that is literally, scientists believe, the limit that this structure, but you could look at a heart as a mechanical device, you can't get it to pump any faster than 1,250 times per minute. So the only other way to supply a tremendous amount of blood to these muscles in the wings is to have the heart be larger. And so if you were to compare a hummingbird heart to a blue whale heart, relatively speaking, the hummingbird heart is 8 times larger than a blue whale heart. And blue whale doesn't have those high energy requirements. As I said, it can beat its heart maybe 10, 15 times per minute. And when it dives deep, much slower than that. So there's tremendous amount of variation here. Just looking at these vessels, some of these blood vessels, they still don't know what they do. So there's a tremendous amount of research that still needs to be done by these folks up at the ROM. This is an exhibit right now. You can go up and see this heart up in Toronto. All right, so I want to move on to horseshoe crabs. This is another, to me, it was a real fascinating adventure. And here, we've all seen these, especially on the East End in the early summer, late spring. They come out into the shallows and they mate. And the term the term living fossil is thrown around a lot, and in reality, this is a real living fossil, because you can see fossils of these guys that are 500 million years old that look just like a horseshoe crab. So to put that into perspective, that's 250 million years before the first dinosaur walked. You had horseshoe crabs, and so they have gone through some real problems. There are 4 different species, and things have gotten problematic for them lately. I was interested in them initially because their heart is very different than ours. If you look at the human heart, as I said, it's got this pacemaker that sends out an electrical signal, and that pacemaker is inside the heart. And in the wake of this electrical signal, the heart contracts, the muscle contracts. Horseshoe crabs don't have that. They have a nerve, a complex of nerves sitting on top of their heart that send the signal to the heart that makes it contract and dilate, which is the reason sort of in the book I say that, this is why when you see pictures of Aztecs and they're holding up a heart and it's beating and at the top of a pyramid, you never see them holding up a horseshoe crab heart or a lobster heart, because if you sever this heart from its nervous system moorings, it's not going to beat, whereas ours would, or that's why you see the priests up there holding these hearts that are still beating. So I was interested in the hearts initially and how to describe how they work, which is kind of complex, but luckily I had these great figures from Patricia Wynne, who's been working with me on every book and every scientific paper I've ever written. Okay, so the problem is that they've been around for 500 million years, but they are in danger at present. In the early part of the 20th century, and look, you can see these walls here of horseshoe crabs. They were used for fertilizer, and much more recently used as bait by fishermen who used them to put them in traps to catch eels and whelk. So this was a problem and really put a dent in populations around here. About 40 years ago, a new problem popped up for horseshoe crabs. And that was, and it wasn't such a problem for humans, because the researchers found out that there was a substance in horseshoe crab blood that could detect endotoxin. And briefly, endotoxin is produced by bacteria, but it's not like they're releasing it to fight off enemies. Endotoxins are released when you destroy a bacterial cell, like you say you sterilize something and there are bacteria on it. When you burst those bacterial cells, these substances, which we call endotoxin, are released. They're really part of the cell membrane of the bacteria. And so the problem is that when this stuff gets into our circulatory system, it can cause really serious problems. Endotoxins can kill you. So the researchers found out that there was a substance in horseshoe crab blood that, when it came into contact with these endotoxins, gave a visual signal. It formed a clot. And so literally an industry was born around this. And you will often see, if you're driving around on the East End, pickup trucks full of horseshoe crabs that are on the way to a facility like this, where they're hung up, drained of blood. And you can see they have blue blood, which is something that you'll find out about why it looks blue in my book. But you're supposed to return these things to the wild by law. But the problem is that you can see here they stuck a cannula right into the region where the heart is to drain that blood. And if you look back here, if you stick a needle into this heart, you can destroy it. So a lot of these animals die. They've lost a tremendous amount of blood when they're returned to the wild. And so this is a problem. Now, the good news, there's good news and bad news. The bad news is that horseshoe crabs are now in a situation where they may go extinct, at least in localized populations especially along the Atlantic. That's the bad news. The good news is that there's now been a test that's been developed that does not use horseshoe crab blood to detect endotoxin. And it was just about, it started to be used by drug companies, and then what happened was that COVID came along, and a lot of these facilities fell back on the old tried and true methods, and there were more of these facilities, these companies that produced the old horseshoe crab test than the new stuff. So the new technology got pushed to the back burner. So hopefully when COVID goes away, we can get rid of the horseshoe crab blood test. Now, the other bit of good news is that Cornell Cooperative Extension researchers there have developed a synthetic smell that the scent smells like horseshoe crabs, and they're giving them out to fishermen to put on their bait, which is not horseshoe crab, in order to prevent having to capture these animals and chop them up and stick them in their traps. So there's good news and bad news. Stay tuned. All right, so I wanted to address this question, how did the heart become the seat of the emotions, intellect, and the soul, and all of these terms that you hear about hot-blooded and, cold-blooded and blue-blooded and, well, I could sit here all day and list all of these things that deal with the heart. And so when I was writing a book about cannibalism, and I wanted to determine where that knee-jerk reaction to the term cannibal or cannibalism came from, I initially named the chapter "Blame It on the Greeks." And so when I looked to see where this idea of the heart as the center of the mind and intellect and memory and emotions, that chapter could have been called "Blame It on the Ancient Egyptians," because these guys, and we're talking about at least 1500 BCE here, in places like the papyrus book of the Egyptian Book of the Heart. These folks knew quite a bit about the heart. They looked at it, they realized that it was important, and they didn't think the brain was very important at all. So if they were going to, after you died, they would yank your brain out of your nose with a hook. Meanwhile, they would remove the heart and preserve it, and because supposedly in the afterlife, the heart would be weighed. Here you can see it in its little container against the feather of Mot, and Mot is the goddess of truth and virtue, to determine what was going to happen to you in the afterlife. So from here, Egyptian medical information was held in really high esteem by the Greeks. And so Greek physicians like Hippocrates and philosophers like Aristotle picked up on this idea that the heart was the center of things. They also picked up on the Egyptian medicine, and some of it was wrong. One of the key things that was an Egyptian origin was the fact that that on the arterial side of the body, that was air in the arteries, and there was blood in the veins. We'll get to that in a sec. So from the Greeks, that got passed on to this idea of cardiocentrism, got passed on to the ancient Romans. And that's all well and good, except the fact that a Roman surgeon by the name of Galen, who lived in the 2nd century CE, there are about 3 million words of his that have been translated, and unfortunately, a lot of what he studied was wrong. Here you can see this idea that on the arterial side, it's on the left side of the heart, there's the air inside the vessels mixed with blood that moves from the right side through these invisible pores in the septum that divides the heart. None of this is true. Galen was working under tough conditions. He couldn't dissect humans, but his work was held in high esteem. And after the fall of the Roman Empire, unfortunately, his work was not translated into Latin. It was translated in the early medieval period by Syrian Christian translators who put a sort of Christian slant on things when they did this translation. So the medieval church in Europe saw this translation and thought, well, this sounds great. And so to them, Galen's word became divinely inspired. And these were things like, some of the things that he said that lived on for 1500 years were the fact that there were these 4 humors in the body that had to be balanced, blood being one of them. And this is why you get people being bled and other, and all sorts of ways to balance these humors in order to keep you healthy in body and mind. An example of that, as I mentioned, was bleeding that took place from ancient times on. The idea that you could, for example, if you were going to amputate someone's arm, you would remove the measure of blood that you figured was that filled that arm up to keep that balance. And this carried on for many, many years. Unfortunately, too long. Now, when people started to realize that there were other explanations besides the 4 humors, then leeches sort of fell out of favor. So as I say here, Bogey hated leeches and the African queen, and John Bobbitt, who some of you might recall, loved leeches, and that's because in the 1970s, there was this resurgence of leech use because 3 of them were used as the term medicinal leeches, and they were used in reconstruction surgeries. So if you had something removed, like an ear, or John Wayne Bobbitt, something else, if you reattach that structure, it was difficult to maintain the circulation. You could knit together the arteries, but the veins were so thin and tiny that a lot of times they did not, that you couldn't put them back. So blood would supply, you could supply blood to the structure, but the blood wouldn't drain out. So it would accumulate, then clot, and now you have a problem with this reattached structure. But if you put leeches there, then the leeches sort of act as a venous system. The blood gets pumped to the reattached structure, just like, via the arteries. But then instead of having to depend on the veins, the leeches would draw off that blood, and then new blood would move in. And they had an anticoagulant in their saliva that kept clotting from occurring. So leeches have now made a resurgence as far as medical benefits. But for the most part, what medicine, in a sense, was stagnant for 1,500 years. Now, when did Galen get the boot? In places like China, India, and the Middle East, it happened much earlier than the 17th century. But when you get to the 17th century, and William Harvey, who's an Englishman, determined that this was actually how the circulatory system looked, that there were no invisible pores in the heart, that there was a complete there was a side here that sent blood to the heart, to the lungs and back. This is the pulmonary system, and then the systemic system that pumped blood, oxygenated blood to the body. Once this was determined, then a lot of other improvements sort of fell into place. These were things, and I talk about these much more in depth in the book, of course. The first stethoscope, which was invented in 1816 by a guy who was trying to figure out how to cope with massive tuberculosis outbreak in Paris. And then the first cardiac catheter, which was installed in himself by this guy, Wilhelm Forstmann, in 1928. And that's another incredible story, as he sort of hoodwinked this nurse into thinking that she was going to be his subject, strapped her down in a chair, and then it wound up sticking this thing into himself. Took pictures of this, and then got fired the next day, and wound up in World War II becoming a Nazi, and then after the war a lumberjack, and then finally winning the Nobel Prize down the road. So this is, if you're writing fiction, you couldn't come up with something more a more weird story than this. But this is a way now to deliver substances directly into the heart without cutting into the heart. And nowadays, how to place things like valves, etc. So huge, but this took place first started in 1928. So the book goes into detail about, I was really interested in how the natural world, how animals in nature can help out with regard to medicine. So you have, for example, zebrafish. You can snip off 20% of their heart, which is probably not a really good idea. And we don't really do that much anymore since the time of the gladiators. But if we were to have that type of damage, or even less to the heart, if you have a heart attack and blood is cut off for a while, then the tissue downstream of that clot is going to die. And when it repairs itself, it does not come back as contractile muscle tissue. It comes back as scar tissue. So that's problematic. But why why does that not occur in zebrafish? So we're trying to figure that out. Burmese pythons, which are huge problems in places like the Everglades, when they eat a meal, their heart grows 40% of its normal of its pre-meal size. And we're not talking about pathological enlargement, which is bad, but healthy enlargement of the heart. How can we allow people who can't exercise after a heart attack or some type of a cardiac condition, how do we get their heart to grow if they're not going to be able to go on an exercise regime? Here, I talk about the Antarctic Icefish with antifreeze in its veins. And right now that they're using that antifreeze in companies to if you ever see ice cream, if it melts and then refreezes, sometimes it's crystallized and it has bad mouthfeel. So they're using this this substance, this very substance, this antifreeze, to keep large crystals from forming in ice cream. And then there's the Takotsubo syndrome. In the 1990s, these Japanese researchers noticed that there were women, mostly women, were showing up at hospitals exhibiting symptoms of a heart attack. But when they checked them out, they found out that their hearts seemed to be perfectly fine. There were no blockages in any of the arteries. And when they put them, when they did something called a ventriculogram, which looks at the ventricle as it's contracting and relaxing, it had a weird shape. It was sort of narrow at the top and round at the bottom, and it reminded them of the traps that the fishermen used to catch octopuses. And so they found out that 90% of these women were postmenopausal and they had had some type of emotional trauma recently. And so this became known in the West as broken heart syndrome. And the idea here, very briefly, is that when your emotions are high, if you've had some type of a horrible emotional event, which these women had, then your sympathetic nervous system kicks in, your fight or flight, and all of these sort of stress hormones are dumped into your body to sort of to speed up your heart rate, to speed up your respiratory system. But it also can aggravate tissues and cause irritation if that happens prolonged. And so what we think is happening here is that normally when your emotions subside, then this system shuts itself down. But in the case of these people who have broken heart syndrome, this system of pouring stress hormones into the blood just keeps going and going and going. The good news is that in 3 to 6 months, it seems to resolve itself. Much more on that in the book. The last thing I want to talk about is here, what's taking place in the future. And I was actually literally blown away when I went to Harvard and talked to Dr. Harold Ott. And he stressed the fact that there are that a lot of people die, thousands of people die every year waiting for a transplant, and not necessarily heart, but liver and kidneys. And so his idea here, and the reason for that is that you've got to have the right tissue type, the right blood type. You've got to carry, you've got to transport these organs sometimes across country. You've got to keep them really cold. This is difficult stuff. So his idea is that instead of using waiting around and having people die waiting for hearts, to build your own heart. And he does that right now with cadavers. And how he does this, and you can see this here is, so here's initially you take a heart, and if you look at it, this is all the muscle. Looking at it microscopically, and they put it through a drain. They hook a drain on it, and they put detergent through it that washes away. And you can see there's less muscle tissue here. It dissolves away the muscle tissue, and you can see that more and more here, and leaves at the end of this something that looks ghost white, but doesn't have any of the cells that your body would reject if it came into contact with. It's just sort of a scaffold, a model of what the heart looks like. This is all connective tissue. So his idea is he wants to take cells from a recipient who'd be receiving a heart, convert them into stem cells. And we have the technology now to convert stem cells into any types of cells we want. Okay, well, now you're going to convert those stem cells into muscle cells, cardiac muscle cells, and then seed them, embed them in a model like this, and literally grow a heart with cells from the recipient. So I asked him, how long do you think this is going to take? And he said he wouldn't be surprised if something like this is up and running in about 10 years. So these are the types of amazing things that I was able to see. So here, just in 30 seconds or less here, I pointed out that well, what would you do about blood vessels on the heart? And he said, well, they're coming. You need to talk to my friend and coworker Glenn Gaudet. So I went to Worcester Polytech, and he pulled out a plate full of spinach, and I said, oh, okay. And he said, well, what we're doing here is that it's very difficult to, as I mentioned before, to transplant veins, especially really tiny ones. So if you look at this spinach leaf, it does have veins, and they do move liquid. And he is running those leaves, a spinach leaf, through that same detergent that we saw before, washing away all of the cellular material, leaving behind just this cellulose, which is what makes this vessel. Your body is not going to reject it. He's going to embed blood vessel cells into the structure and then use them in humans. So this is the type of amazing stuff that is in this book, and that I was blown away when I got to deal with it. Okay, so conclusions. a variety of structures have evolved to address the problem of supplying oxygen and nutrients to bodies of every size. There's a tremendous amount of variation out there. The ancient Egyptians are probably the first to come up with the idea of the heart as this central organ involved in things like intelligence and emotion, and what we would call the soul. And this, this is 3 books that I've written, and in all of them, this idea of Galen and how his teachings really put a stop to medical advancements for something like 1,500 years. And it really, to me, is a warning against the lockstep adherence to any type of idea that and not challenging ideas. That's just not what science does. And then, of course, and I probably mentioned half of them, is that medical advances in the past, present, and future have and will continue to transform cardiac medicine. And I think that's shown really well with Dr. Gaudet and Dr. Ott. So I want to acknowledge the East Hampton Library for having me here. It's great to do a local talk, and to LIU Post, where I was just granted emeritus status. Which means that they don't kick me out if I show up there now. And in the American Museum of Natural History, where I was a grad student and a postdoc, and I've been a research associate there for years. I want to thank my editors. This should say Amy, not Annie, and my publicist Amanda. She's Amy. And Algonquin Books, of course. And finally, I've been working with Patricia Wynne for about 30 years now, and she's absolutely amazing. She's done about 250 books, and I'm just so incredibly lucky to work with her. So if you want to see more of her artwork and perhaps purchase art, you can find her at patriciawynne.com. This was from the Cannibal book that came out in 2017. And if you want to see my stuff, you can look at my website, billschutt.com. I'm on Twitter, Bill Schutt Books, Facebook, and 3 TED Ed videos. If you just look for transfusions or cannibalism, you can find me there. And once again, I hope you've hung in there and that I didn't run too much overtime. And if you have questions, I hope to be able to answer them.
Bill, thank you so much. We do have I see a question in our Q&A now. I just want to let you know the time is 6:47. So if you all have any other questions for Bill, put them in the chat box before we end, and we will get to them as they come. Before we get to our first question, though, I did want to mention that Bill's book is available wherever books are sold. So if you want to buy a copy, definitely do. And we will have copies available at the library. They're making their way to us right now. So our first question is from Kathy. She said, love the reference to John Bobbitt. Is cadaver research for heart transplants widely accepted? Are there challenges to this type of research?
Yeah, these would be donations. It's not like we're dealing with a really good question, Kathy. So we're not talking about grave diggers here. We're talking about people who would donate their heart to science. So rather than wind up in maybe in a medical school, a certain percentage of those hearts would be donated to a system that doesn't exist yet, where your heart would go through this process and be used to transplant into patients who are perhaps on a waiting list. Now, that technology, as I said, is probably 10 years off, and of course they're going to run into problems, and that will need to be addressed. But I think I think that it's a really positive step, especially when you see the fact that they are, that this technology it looks like they're moving along quicker than they thought they might have a couple of years ago. And I'll leave it at that.
Rashni just said, thank you for an absolutely amazing presentation. Janine asks, since an insect has several hearts, some being in the leg or antenna, what happens if they lose a leg or antenna?
Great question. So I believe, and I'm not an entomologist, that depending on what the structure is, that they could survive without a leg that would close off. There would be clotting agents that would work just like in mammals. But if it was, it was something that was, if they lost 3 legs, then they probably would be eaten pretty quickly. So I just think it's a matter of what the structure is that they lose. But don't think of them as hearts. You think of them, and these are not card carrying hearts. They're hearts with quotation marks around them. They're little muscular pumps. And so I made a note of this in in the book that there's not a great deal that's known about them. So it's I put a little alert that it's a perfect place for grad students to move in and learn more about what these antennal hearts are and heart legs are and how they work, because there's quite a bit to learn about them.
Okay, another question from Roshni. Is anyone using CRISPR technology in cardiac research of interest?
They might be, but I don't know. I don't know of any of that going on right now. I certainly wouldn't be surprised to see them trying to splice out, to cut out genes that are identified with congenital heart problems. But nothing that I could point to right now.
Okay, I think that covers it for our questions. That's all I see here in our chat. Somebody else mentioned, oh, Dawn said, excellent presentation. Oh, Kathy also mentioned, I was interested to learn that horseshoe crab scent can be used to substitute by fishermen. Do you know when this occurred?
It's been I think it's recent, because somebody just told me about it the other day. It's not in the book, but somebody told me that at Cornell Cooperative Extension, they were handing out this scent to eel and to whelk fishermen. So I need to learn more about it, but it was really news to me last week. But I'm always looking for sort of good news for horseshoe crabs, and that fit the bill.
All right. Thank you so much, Bill. I really, really appreciate you on behalf of the East Hampton Library. Thank you for being here. And thank you to all of our participants. Thank you, guys.
It was a pleasure. Thank you very much for having me on. Take care, everybody. And thanks for putting up with me.
Have a good night.